fluorescent in situ hybridizations or fish. Definition. Fish is a cytogenetic technique that uses fluorescence probes that bind to only those parts of the chromosome with high degree of sequence complementarity. What does it use for? Fish is used to specific localizations of genes and direct visualizations of chromosomal abnormalities at the molecular level. What does it detect? Fish detect DNA genes, viral DNA, RNA transcripts of mRNA, as well as viral RNA. Probe. Probe is a DNA sequence which is complementary to the target sequence and will bind to it. Probes are tagged with fluorescent dye. Size range from 20 to 40 base pair to 1000 base pair. Types of sample used for fish. There are two types. First one is fixed cell suspensions, and another one is formalin fixed paraffin embedded tissue. Diagnostic applications of fish. Prenatal diagnosis, cancer diagnosis, molecular cytogenetic of birth defects and mental retardations, the identifications of specific chromosome abnormalities, the characterizations of marker chromosomes, interface fish for specific abnormalities in case of failed cytogenetic, and monitoring the success of bone marrow transplantation. Overview of fish. First, prepare the fluorescent probe. After that, the nature of probes and the target. After that, hybridizations of the target, and lastly, detections. Multiplex fish or M-fish and spectral karyotyping or SCAI. Both M-fish and SCAI have the same experimental procedure, but the only difference is the way of viewing the image of chromosomes. Definition of M-fish and SCAI. M-fish and SCAI are a 24-color, multi-chromosomal painting assay that allows the visualization of all human chromosomes in one experiment. What it detects. First, microscopic genomic abnormalities. Second, balanced and unbalanced translocations. And third, aneuploidies. What it doesn't detect. First, submicroscopic abnormalities that less than 5 mb. Second, inversions. And third, duplications or deletions. When to use SCAI or M-fish. It is used as an adjunct to G-banded karyotyping to define complex rearrangement and to identify marker chromosomes. The experimental procedures for M-fish and SCAI. First, all 24 commercially available probes are differently labeled and hybridized to metaphase spread for 24 to 48 hours. Second, post-hybridization washes. And third, antibody detection if required. The differences between M-fish and SCAI.
And is to identify the differentiation of the chromosomes based on that chromosomes present or absent when visualized with specific filters, while Sky is for identify the differentiation of the chromosomes based on their spectral properties.
What is comparative genomic hybridization? Comparative genomic hybridization is a molecular cytogenetic method used for analyzing copy number variations relative to ploidy level in the DNA of test sample compared to a reference sample without the need for culturing cells. C and V is defined, in which the sections of the genome are repeated and the number of repeats in the genome varies between individuals in the human population. Is a type of structural variation, specifically a type of duplication or deletion event. Methodology of CGH. The first step is the DNA labeling of each DNA sample with different color of fluorophore. The test sample and reference sample are then mixed at the ratio of 1:1. And then for the second step is the hybridization of the mixture into a metaphase chromosome. The third step is by using a fluorescent microscope and computer software, the differentially colored fluorescence signals are then compared along the length of each chromosome for identification of chromosomal difference between the two sources. A higher intensity of test samples in a specific region of chromosome indicates the gain of material, while a lower intensity of the reference sample indicates the loss of material in the test sample in that specific region. A neutral color indicates no difference between the two samples in that location. Next, we move on to ACGH. So what is array comparative genomic hybridization? Array comparative genomic hybridization has the same principle like comparative genomic hybridization, but instead of using metaphase spread, ACGH uses array as a site of hybridization. For the methodology of ACGH is the same like CGH.
Okay, now let's move to the prenatal diagnostic and cytogenetics. So what it is? It is the science of identifying structural or functional abnormalities birth defect in the fetus. For example, trisomy 21, trisomy 13, or trisomy 18 and other abnormalities. So why it is important? Because all pregnant women are at risk of carrying fetus with genetic abnormalities. So, what are the purpose of doing this diagnostic? So first, as the early assurance to the at-risk families if the results are normal. Second, in order to allow the couple to prepare for the birth of an affected child. And the last one, as there is information to couple who would not have child without defect. So, what are the techniques involved? It can be either non-invasive, invasive, are minimally invasive. So, for the non-invasive testing, it doesn't require the introductions of instruments into the body, for example, via ultrasound or magnetic resonance imaging or MRI. Meanwhile, for the invasive testing, it will involve the introductions of instruments or other objects into the body or the body cavity either via chorionic villi sampling or CVS, amniocentesis, or chordosynthesis, and it will next be proceed with the karyotyping. And the last one is the minimal invasive testing, which is it will be performed through the tiny incisions instead of the large opening by using maternal blood or cell-free fetal DNA in order to identify the abnormality that occur in the fetus.
Moving on to constitutional cytogenetic and genomic imprinting. Constitutional cytogenetic can be defined as research on genetic abnormalities that are present at birth. Identification of chromosomal aberrations can be done in clinical laboratory testing, which includes sample types of peripheral blood, amniotic fluid, chorionic villi sampling, product of conception, and also skin biopsy. Genomic imprinting. Genomic imprinting could be defined as the epigenetic phenomenon by which certain genes are expressed in a parent of origin specific manner. If the allele inherited from the father is imprinted, it is thereby silenced and only the allele from the mother is expressed. Meanwhile, if the allele from the mother is imprinted, then only the allele from the father is expressed.
Epigenetic modification brings about lasting changes in gene expression. Let's have a closer look. Here is a normal human cell, and we are pulling out its genetic material. The DNA is wrapped around nucleosomes, which are made up of histone proteins. Coming off the histone proteins, we have histone tails. DNA is made up of a combination of 4 nucleotides: G for guanine, C for cytosine, A for adenine, and T for thymine. There are 2 main epigenetic modifications. The first one we will talk about is DNA methylation, where a methyl group is added directly to a cytosine residue that exists in a cytosine guanine sequence, or CpG sequence for short. So for example, here we have many CpG sites that make up a CpG island. And here the cytosine are methylated. The methylation of CpG sites in promoter regions is associated with gene silencing. The second type of epigenetic modification is histone modifications. This is where acetyl or methyl groups are directly added onto histone tails, and this will modify gene expression. For example, histone 3K9 acetylation correlates with transcription activation, and histone 3K27 trimethylation with transcription repression. Genome-wide patterns of DNA and histone modifications, or epigenome, are established during early development and are maintained during cell division. In cancer, these patterns are altered and disrupted.
Human disease involving genomic imprinting include Angelman syndrome, which could cause maternal deletion or paternal trisomy. Next is the Prader-Willi syndrome, which is caused by paternal deletion or maternal trisomy. Here is a picture of each syndrome.